today the real focus is going to be on that done-for-you gene-to-protein service offering. So this is just an overview of what we're able to offer at GeneArt, and the areas that you see in red are really what comprise a lot of our gene-to-protein offering. So we will help you with optimizing that gene to get the highest expression possible. We will synthesize those genes for you, we will clone those, and we will get those into the system for expression, and we can do the purification for you as well, and provide you that protein that you need for your downstream steps. Here are a few examples of applications for synthetic genes and proteins. So whether it's basic research, you need functional assays, you're developing an antibody therapeutic, you're doing any sort of biologics research, the list really goes on and on. Whether it's protein engineering, antibody optimization, if you're doing any crystallization or structural analysis and you don't really want to do all of that upstream work in-house, if you're developing a gene therapy, you're doing any sort of early work around cell line development, or if you need to do any drug and target validation as well. These are all great ways that you can actually leverage the service that we offer to help your research. Our gene-to-protein service leverages our extensive experience and provides quite a bit of flexibility. So we do have a soup to nuts offering that's all in-house. So as I mentioned previously, we can go from helping you take your sequence of interest and deliver that protein that you're looking for. We've developed a proprietary line of cells that we actually use for this process that we are also able to provide as a standalone catalog offering on our XP293 and XP-CHO systems. And those have been developed to really be reliable in our hands as well as yours. We're able to help you with scale and provide additional flexibility. So when you're working in the lab, whatever scale you've already built in for in-house, with those cell lines that I've mentioned, we're actually able to help you with scaling, whether it's you need to do some early work with, say, a 35 milliliter pilot study, all the way up to if you do have particular targets that you're looking at scaling and you don't have that capacity in-house, we can help you with up to 25 liters of volume, of expression volume that we're able to provide there as well. And this all comes with extensive experience in the industry. We've partnered with companies large and small across the industry, including having service agreements with a lot of these customers, and have provided excellent service to these customers as well. This whole process really starts off with our next generation gene synthesis platform. So we use our gene optimization bioinformatics suite to design essentially what we're going to use. We use our gene optimization and bioinformatics suite to actually design the sequence that we will be using for expression. We will then synthesize the oligos that will be needed, assemble the genes for you, do the cloning, do the sequencing, provide final documentation on those as part of the project, and then use those same products in the downstream steps. So from the bioinformatics standpoint, the main crux of this whole process is really on our GeneArt Gene Optimizer software, which takes into account over 20 different parameters to provide that optimized gene for optimal expression. And this goes well beyond just codon usage tables. We look at GC content, we look at cryptic splice sites, we look at a large array of unwanted motifs that can lead to secondary structures, that can lead to truncations or just non-optimal expression levels. So just to give you an example of how this process works, so if we had a sequence of amino acids, which in this case spell out LifeTech, as you can see here, we would have a different mix of what the codons are that are actually available, because the codons that are existent in the natural occurring sequence, or if you're providing an amino acid sequence, we don't have that sequence there. Those aren't really geared towards providing the optimal expression within the system. So what our algorithm actually does is we go through and look at that initial sequence, which may have some that are really non-optimized and have some that are, and we really want to get kind of as many of these to be this green color as possible, but that may not actually be possible, because if they're all green, we may have repeats, or that may create secondary structures, things like that, that will also impact how well your gene of interest actually expresses. So with our algorithm, we look at the sliding window in which we look at a particular set of genes that kind of move from 5' to 3', and we'll go through and optimize based on not just the codon of interest that the software is actually putting in place, but also what's going on around it. And we go through this a few times to actually get to the right set of codons to give you that optimized sequence that should give you the optimal expression. So this isn't just theory. We actually know this works. Here's some data from a peer-reviewed paper that compared our GeneArt algorithm to a number of competitors in the market. As you see consistently, the GeneArt algorithm outperforms many of those on the market. Part of the core of our service offering here, and part of what makes us different than any other provider for a protein production service, is this ability for us to not just do the service for you at our facility, but our ability to enable you to continue to work with what we've provided downstream. And what I mean by that is we use the same process for production at our facility as we recommend for anyone who's using our XP293 or XP-CHO expression systems. And those are available commercially as part of our Gibco line of products. Our primary offering is really around insect cells and mammalian cells within our GeneArt facility. And as I mentioned earlier, we do have a lot of flexibility with regard to scale. We can do high throughput small scales for you, and we can work all the way up to liter volumes as needed as well. And our capabilities don't just stop at expression. We also have a full suite of purification ability as well. We can do affinity purification followed by ion exchange or size exclusion chromatography. We were able to do affinity chromatography with any of the most common affinity tags, so those things like his tags, FC tags, strep tags, flag tags, etc. And then we also do have additional options for any polishing steps. We can do protease cleavage if needed. We have aliquoting services. We can normalize your concentrations for you as well. So we do have a full service offering with regard to our purification service methods as well. And to support that, we also have a full set of analytical capabilities. And this is really so that we can provide you any characterization information that you need, as well just with regard to what we're sending you. So we have a number of things that are included, like STS page and Western blotting. We can measure, and as I mentioned, normalize for protein concentration. In addition to providing a preparative size-choosing chromatography, we can do analytical size-choosing chromatography. So if you do need to see that your protein of interest is in a certain monomeric or multimeric state, we can also do endotoxin determination. So if your protein is actually going to be used in vivo study downstream, we can provide you analysis on what the endotoxin levels are there as well. And then if you do have any specialized analytics as well, there are cases where we can bring those on board. So please inquire if you do have anything specialized that you would like us to look into. So this is just an example of really how our service is able to help you kind of in live action, so to speak. Or here's a live example of how this can work. So this is a case study of really how we were able to enable a customer to not just save money, but also save time by leveraging our service. So in this example, we have this biotech that was looking to produce a biologic. And initially they were using our XP293 system in-house, but they hadn't really done a lot of the optimization steps up front. And their two main goals were that they wanted to increase the amount of yield they were getting, or so essentially they want to get more bang for their buck with regard to expression in purified protein, but they also wanted to reduce the number of hours that they had because resources were relatively strapped. So what we did was we helped them with transitioning from their XP293 system to our XP-CHO system, which does provide a higher level of expression. And while that produces higher yield, they still were doing a lot of this in-house, so you still had this high level of full-time employee or FTE utilization to perform a lot of the downstream steps. So once it's expressed, you still have to purify it and things along those lines. So we continue to work with this customer and introduced our gene to protein service, which is performed at our GeneArt facility. And with that, what we were able to do was provide the customer a highly purified product, and we're able to provide them with a saving of over 2,000 hours of work just by helping them with that transition from their old system to being able to use our services to help them, enable them to do more in less time. So just kind of in closing, why partner with Thermo Fisher? So first of all, you get all the benefits of outsourcing, but with the ability to have that backed up with commercially available reagents. So this is not a scenario where you're outsourcing the product and then you have to then go and re-optimize everything to bring it in-house. As I mentioned, we do the same process at our facility as we recommend for our commercially available reagents, which makes it very flexible and very easy for you to start work, which you then send to us, or have us do initial work for you that then you continue on in-house. We have extensive expertise in the full workflow, all the way from gene optimization through to expression and purification. We've developed specific sets of cell lines that are optimized for purpose, in this case, for protein expression. We have a full suite of flexible solutions, everything from helping you do expression and purification for high throughput or screening types of applications, through to being able to do liter scale scale-ups, which gives you a lot of options as far as how you can adapt your workflow with our offering. And we have a full team that can keep you in the loop with regard to what's going on with your project. So we have dedicated teams that will help you set up the project, that will be very transparent as to how the project is progressing, as well as documentation to show you exactly what's being provided. And then we are able to customize purification schemes if as needed for your particular application. At Thermo Fisher, we are very committed to our mission of helping our customers make the world healthier, safer, and cleaner. And a great example of that is that our facility at Regensburg, where our GeneArt services are performed, is a zero waste certified manufacturing facility. And that is that 0% of the waste we produce at our Regensburg facility goes to landfill. So not only are we able to provide you with great quality product, but we're also able to do this without adding to waste in landfills as well. So ways of getting in contact with us or learning more. You can visit our website at thermofisher.com/GeneArt. Contact your local sales representative. If you're in the state of Massachusetts, that would be me. And you can also feel free to email us at geneartsupport@thermofisher.com. Thank you for your time.